What is gene editing and how does it work? As we saw in the previous video, it's a very disruptive technology. And on this slide, I wanted to list the main takeaways from the video and the most important things for you to learn and understand about this technology. In general, gene editing is based on the discovery and research of the CRISPR sequences in the immune system of various bacteria. The two scientists, Emmanuel Charpentier and Jennifer Doudna, published a paper around this in 2012 and got the Nobel Prize of Chemistry in 2020 for their work in the field. These two scientists basically discovered the CRISPR or invented the CRISPR-Cas9 technology, which is often called the gene scissor, like you can see on the picture on the left side. So with this technology, you can do surgery and edit genes to either change, delete, or add DNA to any organism. So this technology can be applied to humans, to animals, and to plants, as we will learn later on in this module. With gene editing, this technology is converging with AI, with artificial intelligence, because if you look at the human genome, these are huge data sets, and you need AI algorithms to find out in a fast way which sequences and which strings of the DNA you want to alter, because it would just take too long for a human to read every line and then decide which string to alter here. Another thing with gene editing is that it's pretty simple and cheap to use. So you have this like democratizing of biotech, and there are companies who are selling like products for hobbyists on the internet. So this is something which could be of concern, and we need to really see how the usage of this technology is evolving. And there is, in some cases, more regulation needed. One very important thing for you to take away as well is that scientists currently focus on somatic cell editing due to the uncertainties around germline editing. What's the difference between the two of them? Somatic cell editing means that you edit the DNA of an individual, but only the individual is affected by the consequences of editing his or her genes. While with germline editing, you also edit eggs and sperms of the individual, so he or she can pass it on or pass it down to successors, and the consequences of this are not fully understood yet. So in the scientific community, there is consensus around this, that for the time being, it's better to exclusively focus on somatic cell editing.